Hi, everyone. My name is Jason Key. I'm SP Grid, Harvard Medical School in Boston. Thank you for joining us today. Today we have Christian Olsen joining us from Research Triangle Park in North Carolina to tell us about Geneious. Feel free to send questions to me during the presentation in the chat window. Chat window is here. It has the little caption bubble down at the bottom. And I'll pass those on, or we can save those to the question period at the end. With that, Christian, are you there?
Yes, I'm here, Jason.
Yeah. Great. Go right ahead.
Awesome. Thank you so much, Jason, for having me. And thank you, everyone, for joining us for this small portion of this fantastic lunch break series. I'm really thankful to be a part of it. As Jason mentioned, my name is Christian Olsen. I'm a field application scientist for Biomatters, which is based in New Zealand. And our flagship product is called Geneious, which is what we're here to learn a little bit of, which has been around for about 5 years now. I should say that major releases we schedule about annually, and we're looking for this October to be our version 10 release. So stay tuned for some interesting things to happen this fall. So what I need to do is I need to give a little bit of a background. And in terms of how the software works and in terms of orientation, we'll get into briefly, but I wanted to provide an agenda for what we'll be talking about. And it's a little bit of a variety, and we do have a lot of material to cover. So bear with me as I move through this, and I'm happy to entertain questions afterwards. I'll also provide my email address for those people who have more in-depth questions that need to be answered. So a bit of an introduction. We'll be speaking about Custom Blast. We'll highlight our workflow builder, which is a massive time saver for people working in the sequence analysis space. I'll talk about a number of annotation tools, and finally I'll finish up on one of many in silico cloning methods within the software. Probably one of the most famous right now, anyway, for Crispr. Okay, so to let everyone know where we're coming from, we design Geneious for biologists, and we do this to free up time and resources for bioinformaticians. We want to make it easy for biologists to analyze their own data, to visualize whatever sequence data they have. And with this, we're looking to extend time as much as possible. We also, within the sequence analysis space, want to work with small volumes of data or large volumes of data. And so it's with this end in mind, we are seeking to make what I think is the best sequence analysis software out there. So Geneious will work on whatever operating system you have, whether you're a Mac user or Linux or Windows PC. Geneious will run on all of them. It shouldn't matter to the user what operating system you have. Geneious should run, and it does, and it runs quite well. Recently, we did a lit search on Google Scholar. We found that our software is the most cited commercially available software available. And this is a really nice testimony for user adoption and just where and how much it's being used. So quickly, a tour of the interface for those of you who might be new to Geneious. When you install Geneious on your computer and you open it up, this is what you see. It's a fairly standard orientation for most software packages. On the left-hand side is the sources panel. And the sources panel is nothing more than different sources for your sequence data. Okay, so at the very top, we've got the local directory. And then down below, we have live sources like UPathDB, NCBI, and Uniprot. And those are all various places, not only where you can retrieve data, but where you can store data. Okay. So the local directory is actually where your data live on your local machine. This is, I differentiate this from a shared database where you have a shared data resource that a number of users hook into to collaborate with. Okay. So down below, The top icon is that shared databases feature I mentioned. Whatever relational database you have, Geneious can hook into. But you also have these other resources like NCBI. So if you wanted to download a genome, the wonderful thing about this is you can download whatever genome is available on NCBI, as well as all of the metadata that are associated with that genome. Now at the top center is what I call the document table. Okay, so we call sequence files documents or sequence documents. And you might hear me interchange these terms frequently. But when I click on a folder in my local directory, the contents of the folder is displayed in this area right here. Okay, so we have a number of different elements that I'll just speak to real quick. I've got icons to the left of the names of the sequence documents. I've got a description, as well as some other column headers, which can be grabbed with the mouse, moved around. And so this is a nice, fast way to look at your sequence data within the folder. Now, when I select a sequence document, the center panel becomes active. And right there in the middle, where you'll see a list of eukaryotic species, that's the sequence viewer, which is interactive. And that shows one way of visualizing your data. Okay, so you see I have a number of tabs that run across the top. Here I've got the alignment view showing, but I can click on the annotations tab to look at just the annotations view. I can click on the distances tab to look at a matrix of distances. I've also got some other tabs that I'll leverage a little bit later on in the presentation. Just to the right of the sequence viewer, you'll notice the options panel. And here I've got consensus box checked. I've got a highlighting option checked. You'll notice a number of tabs over there to the right that run vertically. I've got a little home tab at the top, the display tab, which I have shown here. These are different options that you can select or deselect to change what you see in the sequence viewer. So I just think about these as different lenses. Okay, so if I would like to look at various graphs or annotations, I can select those from the tabs that run vertically. Over there to the right, you'll notice that I've got a help and tutorial panel. Okay, the help and tutorial panel is a series of self-help tutorials for self-guided study. So if you're a PI of a lab and you've got 6 or 7 postdocs, you can direct them to this panel to learn various analyses in Geneious, how to create a multiple sequence alignment or a BLAST database or how to design primers. If you've forgotten something, this is a great resource. As you're learning, you'll remember more and more, but every now and then you forget where something is. You can go to this help and tutorial panel to jog your memory. And then at the very top is the menu bar. Okay, the menu bar is one of the main ways you'll interact with Geneious. And the logic is that you'll select a sequence document, you will go up to the menu bar and you'll select some type of operation. And we try to bin them according to common features. So all alignment and assembly operations are underneath align and assemble. All of the tree building methods are underneath tree, and so on. Now, remember a few moments ago I mentioned the help and tutorial panel to the right. If I select that orange question mark at the top, that will open up the help and tutorial panel. If I select it again, it'll hide it. So if you're concerned about screen real estate, that's one way you can shrink or expand panels in Geneious. We've also got this blue support button, which I think is one of the most critical buttons in Geneious. If you ever have any questions, please select that support button. That'll create a ticket, a support ticket, which will be sent to our support desk. Most of the time I get copied on those tickets, so if it's a critical situation, a triage situation, that I can step in and help. And since I'm on the East Coast, as you all are, I can help pretty fast. So please use that support button. As well, the green backup button to the left of that, that makes it very easy for users to back their data up. And I cannot stress this more. Please get on a backup schedule. This is probably the #1 support ticket we get If a database corrupts, which happens, and you've not backed up your database, then you're stuck. And so if you can set a calendar alert to remind you to back your data up once every 2 weeks or at whatever interval you feel best, please use that backup button. Okay. Now, just a few things that I'll speak to before I run into our first topic, which is custom blast. So I've got Geneious running on my machine, which I think you all can see now. Keep in mind that these panels can be shrunk or expanded. Okay, so you can play with this to increase or decrease the screen real estate. The document type that I have selected in my document table is going to dictate which tabs I have that run across the top. Now, this seems intuitive until you're actually in the thick of it and you're tired and you're kind of forgetting what you've done. If I drag and drop a PDF into my document table, it'll import the PDF, but I will not have some of these tabs across the top, like the sequence view and the links graph, because my document type is type PDF, which doesn't have a sequence view. Okay, so you'll notice that these tabs change across the top, and they will change depending upon what sequence document type you have. So in this case, I've got a number of Sanger reads. So of course I have a sequence view. I've got a links graph, which is very nice, a text view of the document, as well as a FASTA nucleotide view and an info view. Okay. Now, the last thing I'll speak about, if you've installed Geneious on your machine for the first time, there are a number of plugins that come installed by default, but the majority do not. Okay, and you can install plugins underneath the Tools tab at the top. So underneath the Tools tab, you'll find the second to the last option is Plugins, and this will open up the plugin manager. Okay, so you see I have a number of plugins already installed, but I've got two that I haven't, like this CPG Island predictor and this collaboration plugin. You can filter on the columns, so you can filter according to category or type or description. We have a number of plugins for assemblers, tree building methods, annotation tools. So I would encourage you please to go check out the plugins menu and find the relevant plugins for your use case and install them. Okay. I'm kind of shining a little bit of light on this right now because when I talk about workflows later, you're not going to be able to use portions of the workflow if you don't have the corresponding plugin installed. Okay? For instance, you won't be able to use the BB mapper in your workflow builder if you don't have the plugin installed. Okay? Same thing for coil prediction and everything else. So make sure to check this out and install them. Okay. Okay, with that in mind, let me go back to my presentation and talk a little bit about custom blast. This was one topic of interest that we received some feedback on. And the first thing you need to do if you want to perform a custom blast search, which I contrast that against the NCBI blast. A custom blast allows the user to define a group of sequences with which they can compare an unknown sequence against. Okay. Now, within Geneious, you can also take an unknown sequence and you can send that sequence to NCBI and you can do a search on those publicly available sequences. But if you're a competitive lab, say you're in the malaria space where everybody is careful about their sequence data, or you've moved on to a pharmaceutical company where you're not allowed to blast through the firewall, oftentimes you'll have a custom blast set up. Okay, and in Geneious, it's trivial to do. Now, the first thing you need to do is you need to set up blast services. So with the fresh install of Geneious, you cannot set up a custom blast database until you download the blast protocol from NCBI. And that blast protocol, if you can go up to Tools, the Tools tab at the top, and you'll select Add Remove Databases, and you'll see another pop-up window that opens up, and you're going to choose the last option, which is Set Up Blast Services. This is one of the more common things that people forget, especially after a presentation about custom blast. They forget this pretty critical step, and they run into a roadblock. They can't set up their custom blast database. You only have to do this once, and you have to do this once on every machine that you install Geneious on. So if you have 10 laptops in your lab and you install it on one, you have to install the custom BLAST protocol on the other 9. Okay. So depending on your internet connection will download it, I'm sure, relatively fast where you're at. The service that you're going to choose in the setup BLAST services is this custom BLAST service. And Geneious will also show you where the database location is going to be. Okay. Now, to use custom BLAST, this is whenever you download it, Geneious will tell you from which path you are downloading it from, and it will automatically place it in the spot where you need to have it placed. Okay, and it'll give you a status. So in this case, when you download the BLAST protocol, it gives you the version of NCBI BLAST, and it says you've got 7 minutes. So you've got a little time for a cup of coffee. Probably when you come back, it'll be finished. Okay. Now, once this is set up, it is a trivial thing to do to build a custom BLAST database. And I'll just talk about the flow a little bit, and then I'll show you what it looks like to interact with it. So here I have a sequence list of 20 envelope genes, 20 HIV envelope genes, okay? And this is what I want to create my custom BLAST database with, all right? So I'll select the sequence list, I'll go back up to Tools again, and I'll choose Add Remove Databases. And the next thing I'm going to choose is Add Sequence Database, okay? Now Geneious will prompt you to give the database a name. And my recommendation is to give your database a name and attach a date to it. That way you know how fresh it is. As we all know, sequence data get out of date quickly. And when you're managing multiple BLAST databases, you want to have some cue to let you know how old your database is. So here I'm selecting to use the 20 selected sequences in my document table. Okay, now if a colleague emails you or hands you a USB stick with a FASTA file, you can select to create from file on disk. So if you've got 100,000 sequences that you want to create a custom BLAST database with, and it's a FASTA file, then simply select create from file on disk, browse to where that FASTA file is. You'll have to designate what type of FASTA file it is, whether it's nucleotide or amino acid. And then when you click on the OK button, it will create the database. All right? And it will create the database and place the database above the NCBI BLAST resources. Okay, now this may not be obvious, so let me talk to this just for a few moments. Okay, so if I'm going to BLAST against a custom database, here I've got Geneious open, and I've got a phage sequence opened up in my sequence viewer. When I click on the BLAST icon at the top, okay, it'll open up the BLAST dialog box. Okay, and this should be familiar to many of you. The first dropdown is the database that you want to select. Okay, so when I select that, these are the public databases that anyone can BLAST against. But these are the list of all of the custom BLAST databases that I've made. So in this case, I've got Leishmania species DNA custom BLAST database set up on my local machine. I've downloaded all of the Cyclospora sequences as of 12/15/15 from NCBI. I downloaded a version of OrthoMCL, and so on and so forth. Okay, so this is where your custom BLAST databases are going to be listed, and this is where you're going to choose which database you want to use. If you want to use the public resource, the public NCBI resource like NR or EST mouse, or if you want to do PDB sequences, which is another public resource, then just select those from down here. Okay. Now, are there any questions about the custom BLAST database feature that I mentioned so far? If there are, please feel free to ask me, or again, you can save the questions for later. But the critical step for this is to set up BLAST services, which I have highlighted here. Okay, do that once for every machine, and after that, you can BLAST to your heart's content. Okay. If you want to remove a database, which is just as critical. So say your database was just updated, your sequence data have just been updated, you can remove a database, okay? And the remove database is, there's no going back. So once you remove a BLAST database, it's gone. So to remove a custom BLAST database, then you can just select, in this case, I'll select the NCBI Cyclospora data that I have because it's old, and I'll click on the OK button. Geneious will give you a second chance. Are you sure you want to do this? You say yes, delete database, and now it's gone. Okay, and I know it's gone because when I go to look at my database selection, it's not found here in this custom BLAST list. Okay, perfect. In terms of limitations for custom BLAST database, I have pushed this around quite a bit on my local machine. So I'm running a MacBook Pro, 16 GBs of RAM. It's an i7 chip. So I can build a BLAST database of 5 million sequences quite easily, depending upon my unknown sequence length and your hardware resources that will dictate how fast the search happens. Okay, so you may want to make a number of different BLAST databases. Okay, so let's talk a little bit about our workflow builder. Okay, in our workflow builder, we introduced this, I believe we introduced this in version 7. And the workflow builder allows the user to create a workflow with multiple steps and to save that workflow so that they don't need to do those individual steps every time they want to run their sequence analysis pipeline. Okay. So I've got a specific use case that I'll talk about that I helped out with at the CDC. We use the workflow builder for a Plasmodium falciparum sequence analysis workflow. Okay, and the general flow, just to give you an idea as to what we did in this workflow, we identified a gene marker. So in this case, the very well-known K13 gene. We identified and annotated a reference sequence that we included in the workflow. And then within the workflow, we built trimming step, mapping steps, annotations of SNPs. Okay, and it's a fairly small workflow. These can get as complex or as simple as you want. And I would recommend start out simple. And if you need help with workflows, please send me an email and let me know. Sometimes they're intuitive and sometimes they're not, depending upon the person. Okay, so this workflow we built, the K13 gene was qualified in this Nature publication. And in Geneious, which you may or may not know, you can download sequence from NCBI. I mentioned that when you do that, you download the metadata. Okay, so in this case, I've got the K13 CDS annotation, which is on my sequence, and it's shown there in yellow. And then I added custom annotations, which are those green blocks for canonical SNPs. Okay, and these are SNPs that we mined from the literature and we annotated onto our reference sequence. And this custom reference sequence that we made is what we included in the workflow. Okay, so all of those green boxes are SNPs, as well as we've got a way that we named the SNP, which you can see in next to some of the SNPs in green. Okay. So in version 9, which is the current version of Geneious, we added a number of tools that Brian Bushnell developed. One of them is the BB-Duck tool for adapter trimming. And we added this because this BB-Duck tool trims specific adapters from kits. It not only trims the full adapter, but also partial adapters. Okay, and you have a number of options here within this trim using BB-Duck step. Okay, so after the trimming step, there's a map to reference step. Okay, and we've got a number of tools for NGS and Geneious. But remember, we created our reference sequence by annotating canonical SNPs found in publications. That first drop-down menu is the reference sequence that we've designated. Okay, and then we've got a method. So you'll choose your mapper, sensitivity settings, fine tuning, as well as saving results. Okay. When you do a reference mapping, you'll return a contig within Geneious. And you'll notice here in my sequence viewer, I've got my reads that I aligned to my reference sequence. All of my canonical SNPs are over there towards the 3' end in dark green. And you'll notice that you're not able to see the reads because you have to zoom in to see them. But this is what you normally get returned when you do this in a manual fashion. Okay. To find variations in SNPs after the reference mapping, then you have to go into Geneious, into one of the annotation tools to find SNPs. You have to set the appropriate SNP parameters, and then Geneious will go through and annotate SNPs, which are added as a track. Okay. So this is all fairly manual that I've shown you. Now, this is not too bad if you have to do it once in a while. Okay, but we needed to do this across 5,000 patients. Okay, and it's just not scalable to do each of these steps 5,000 times. Okay, and so the workflow builder takes all of the steps that I showed you, the trimming step, the mapping step, the SNP calling step, and packs that into a workflow. Okay, so and I'll talk about how to build a workflow in a little bit, but I'll just speak to how this workflow is built. So we gave the workflow a name. We gave it a description. Okay, so documentation is huge. Make sure you are you're robust enough in your description so your colleagues know what your workflow is attempting to do. For each step, we've got an individual horizontal bar. Okay, so for each document, we are going to trim using bbduck, okay, with certain parameters. Okay, and once that step happens, for each of those documents, we're going to align and assemble and map to reference. Okay, and we're going to have variables that we have assigned for that step. And then for each document, we wrote some custom Java code that you can include in your workflow. From there, Geneious will find variations in SNPs according to the parameters that you preset. And then you're going to save the output of each of those. Okay, so if a colleague creates a workflow for you, perhaps you're collaborating with somebody in Thailand and they've used Geneious to create a workflow and they've sent it to you, you can drag it and drop it into Geneious and run their same workflow. So this really gets to one of the hairballs of sequence analysis, is that now you've got a way to perform a reproducible set of sequence analysis steps. Okay, and one of my pet peeves are when people publish their sequence analysis pipelines in a journal and you can't run their pipeline, right? And it happens all the time. So in this case, if you build your pipeline in Geneious, you can publish this pipeline to Git or to your website. You can use it in your supplementary data. People can drag and drop your pipeline into their copy of Geneious, and now they can do your same sequence analysis. Okay, so if your colleague sends you a workflow and you don't know what choices they made at a certain step, simply double click on a step and you can see what choices they made. Also, when you build a workflow, you have some choices you need to make, such as, I don't want to expose any options. Okay, so if you've got an SOP that you're developing, perhaps it's best that you expose no options after you've validated your pipeline. Okay, once you know your pipeline works, then lock it down. Okay, sometimes you want to give the user some freedom to choose their own reference sequence, maybe, or maybe you completely trust your colleague and you want to expose all of the options. Okay, just know that it is at this point that you can dictate how stringent or how loose your workflow is. So if you want it reproducible, my recommendation is to lock down the options. Okay, and just add enough documentation so that the next user understands what the workflow is going to do. So in this case, this Plasmodium workflow takes a set of paired Illumina reads, okay, and it will, this paired set of reads, I'll select in the sequence table, in the document table, and I'll go up to my workflows icon at the top. Okay, and you'll notice a number of pre-canned workflows that we wrote and included in Geneious that you can explore, you can use, you can extend. In this case, the workflow that we designed was this K13 multiple drug resistance ID multi-sample workflow. Okay, so I selected my raw paired Illumina data, select the workflow, and then I hit the go button, and this is what's returned after all of the steps have been gone through. Okay, so hopefully right off the bat, you can see that we've eliminated a lot of different steps, and each of those steps that we eliminated are opportunities for error to be introduced. Okay, we all get tired when we stare at our screens 10 hours a day, 7 days a week. It's best to automate as much as you can with sequence analysis. Okay, so here we did the trimming, we did the mapping, we did the SNP calling, all in one fell swoop. Okay, and if I zoom in, then I can zoom in and see the canonical SNPs that were annotated on my reference prior, and then I can see the SNPs that were called as part of the workflow. Okay. Now, the results of this particular workflow that we did a study. We were able to take a look at 417 patient samples across 10 different provinces in Thailand that were genotyped for this K13 marker. And we were able to specifically look at artemisinin resistance. We were able to look at the positive selection signatures, as well as confirm or establish a nice case for evidence that artemisinin alleles arose independently across Thailand. Okay, so we actually found 2 new SNPs with this workflow and this data set, okay, which I've got highlighted here in red. But we were able to confirm all 7 of the K13 mutant alleles. We found the most prevalent artemisinin resistance associated K13 mutation, of course, is C580. And we were also able to discern 2 distinct haplotype profiles that were separated according to geography, the Thai-Cambodia, Thai-Myanmar borders. Okay, so it was a fantastic workflow that we made, that we published. We actually sent it to the same workflow as being used in Senegal right now to track artemisinin resistance in Africa. And we've got some interesting data to publish here. It's actually submitted for publication, but with a minor tweak to a couple steps of the workflow, we were able to reuse what we did in Thailand in Senegal. Okay. So the workflow, if you had noticed in Geneious, you may have noticed the workflow icon at the top of the toolbar. If you have Geneious installed, you probably won't see that. And that's because you need to add it to the toolbar. So you'll simply right-click on the toolbar, and a little shortcut menu will pop up. Okay, and this will allow you to customize your toolbar. Okay, perhaps you don't do any cloning or any primer design. You can hide those icons. Or if you'd like to add the workflow icon, you just choose Customize, and at the very bottom, select Workflows. Okay, if you do lots of translation, or if you do lots of structure alignments, or submit sequences to GenBank, you can select those icons or deselect them. Okay, so just one little tip and trick to customize the toolbar in Geneious. Okay, now when I select the icon that I've added to the toolbar, you'll notice that the very top selection is Manage Workflows. Okay, and this gets to the Manage Workflows interface. So here I'm showing all sorts of workflows that were added. I've got a workflow that one Geneious user, David O'Connor, who's a Zika virus researcher, he created and published a Zika workflow in Geneious called Zquencher. Okay, and you can go to his website and download it and run it. Okay, I've got an HIV workflow that I created, as well as a number of others. Okay, so if I'd like to examine a workflow, I can just select it and click on View Edit. Okay, so I'll view the workflow, and here in this case, it's rather boring. It's just a piece of custom Java code that was made. Okay, if I want to look at the Zquencher workflow that David O'Connor made, then I can select View Edit, and I can see it's quite robust. Okay. So it's as robust as you want to make it or as simple as you want to make it. And just to bend your mind a little bit, you can nest workflows within workflows. Okay, so when you do that, I recommend you do that with a colleague. That'll prevent you from losing your mind. That's when it gets a little strange. But at each step, double click on the step, and that will give you the parameters for the step. So it's rather trivial to navigate. If you want to build a workflow, you just choose the first option, add new workflow, and you'll see that this is my blank canvas. So I'll call this my hot workflow, okay? And the description is, seek analysis, for naiad pathogens, A through C. Okay, very generic. You can select the icon that you want to assign to this workflow. You can even choose a custom icon. Okay, so if your lab group had a custom icon assigned to the group, you can assign that to your workflow. The first step you'll do is you'll add a step here on this tab. Now recall when I first started this discussion, I talked about plugins. This is where your plugin options will come into play. So I have almost all the plugins installed. I've got 139 operations to choose from, so I can select that. And I've got a live filter and search. So if I want to do a live search on all of the alignment steps that I could use in my workflow, I just type in alignment in the live search field, and it pulls up all of the alignment steps. Okay, so nucleotide, protein, NGS reads, 3D structures, it doesn't matter. It's all the same type of thing you want to do. It's an alignment process. Okay, so in this case, I'll choose to run a muscle alignment. Okay, so it adds that step to my workflow canvas. Now to set the parameters, as you all know, just double click. I'm going to expose no options. I'm going to set the number of iterations to 6. And for those of you who have used muscle, you know that there are a lot of parameters to muscle, and those are found underneath here, more options. So you can set those parameters, such as the maximum amount of memory you want to allocate. Okay, and then once I do that, I've set that step. Okay, now let's add another step. Okay, so I'm going to save the document, and I'll save it in a subfolder called aligned. Okay, I'll click OK. Okay, so here's my save step. And so you can see you just quickly add steps. Okay, so perhaps I want to build a tree off of this alignment. Okay, so I'll build a tree using PhiML. Okay, so there's my tree building method. Select that. There's my third step. Double click to set the parameters. Okay, so in this case, this doesn't provide opportunities to set those parameters, right? If I have lots of steps and I want to move them around in a gross way, I can simply drag and move the steps around. Okay. So my recommendation is to get on a whiteboard with your colleagues, rough out the steps, and there you can start refining the steps of your workflow. Okay. Underneath this Add Step tab, you'll see a number of other options such as renaming. So you can rename or add a renaming step or add a filter step if you want to filter on length or name. And then here's my favorite, add custom Java code. Okay, so for those of you who are familiar with Java, you can add your Java block here. And if you need help with documentation for this, you can click on the link for the Geneious API. And our Java doc online is very mature. It's public. We invite everybody to dive into this. It's a natural and easy way to extend the functionality of Geneious. Okay. Okay, so to export a workflow. So once you've built your workflow, all you have to do is select the workflow and click on export. And that will export a small file that you can attach in an email. You can upload to GitHub or SourceForge, and you can share it with the community. So we have a number of academics and commercial companies that will set up a workflow group within their organization where people will build NGS workflows and they'll share them and they'll extend them. And it's a really nice way to get a lot of work done in a short amount of time. Okay, so that's a bit about workflows. And I see that my time is running fast. So The next thing I'll touch on are annotation tools within Geneious. And truth be told, I could spend probably the better portion of a day speaking about all of the annotation tools in Geneious. But I want to make you aware, when we designed Geneious, we designed it so that you can visualize your data in a very powerful way. You can create images of your sequence data in an easy way. For the annotation tools that we continue to add, we have a number of tools that you can leverage. We have two gene prediction methods that were written for Geneious, Glimmer and Augustus, depending upon if you've got eukaryotic or prokaryotic sequence data you're working with. We've made it so that it's a trivial thing to do to transfer annotations. So if you have an annotation pipeline that you're wanting to execute, we've made it trivial to do. We've made it very easy to export an annotation table. Okay, so once you have a list of annotations, like a SNP table, most of our users who work with NGS data, they want that SNP table. A lot of our clinical users want the SNP table. We've made it very easy to do that. We made it easy to design custom annotations, okay? And we made it easy to add your own metadata. Okay, so remember back to that falciparum workflow that we designed. If I wanted to add a metadata field like GPS coordinate or mosquito nets, has net or doesn't have net, all of those bits of data that the epi officers really rely upon, you can create custom annotations and custom metadata in Geneious. You can leverage the annotations in Geneious to extract sequences. So you're not just extracting the annotation sequence, but you can extract flanking regions. So 5', 3' flanking regions. If you want 500 base pairs upstream and 300 base pairs downstream, if you want to extract the gene context, you can do that. We've made it so that you can annotate your protein data using InterproScan. And the fellow who wrote our Augustus gene prediction method plugin also wrote our InterproScan plugin. He's a member of the community, Mike Thon, out of Spain. We have a number of ways to create graphs, heat maps, predict open reading frames, compare annotations. If you're running a protein production core, you can optimize codons. So the sky is kind of the limit with our annotation tools. Okay, and just to speak toward those quickly, in this case, so I'm showing just a clone, okay, in Geneious, and I've got a number of annotations that were pre-assigned. If I have a region on my clone, if you're running a cloning lab and you want to annotate a region for some reason, you would simply select the region in the sequence viewer, okay, and click on the Add Annotation tab. Okay, so I'm just going to put custom annotation. Okay, and you can assign the type. So we have a bunch of predefined annotation types like CDS, miscellaneous, motif. I can assign directionality. I can make it undirected if I'd like. Okay, in this case, I'll assign it the forward direction, and I want to change the color. Okay, so I'll just double click on the color box, change it to this turquoise, and click OK, and select OK. So here's my custom annotation that I created with the color that I've assigned. Okay, all of our annotations are found over here to the right underneath the Annotations and Tracks tab, this yellow arrow pointing to the right. Okay, this turquoise annotation was type motif, and it was added to my annotations menu. Okay, so as I move along, I can even select a region within that, and I can add another annotation. I'll do very custom. This is just kind of a cartoonish example. So I've added a sub annotation within my custom annotation region. Okay, and so now I've got two motifs that were added. Now notice in the Annotations and Tracks window, if I select or deselect, that turns on or turns off the annotation view in my sequence viewer. Okay. Also notice in the Annotations and Tracks panel, I've got this little Excel spreadsheet over here to the right. And as I select a cell, it will hop to that annotation in my sequence viewer. Okay. Now I can also pop this annotations table out by selecting the pop out button. Okay, and this is where your annotations table can be leveraged quite well. Okay, so I can organize my annotations table according to the way that makes sense to me. So if I've got a SNP table that I want to organize, I can simply move the columns around. This is a rather boring view. So if I want to add more columns, I can click on the columns button, and I have a number of other options like %GC. I can add created by. Okay, I can add NCB feature key. Okay, if I've downloaded the sequence data from NCBI, I've got a feature key that was assigned. Okay, I can also add sequence name. Okay, so there are a number of elements that you can add here. When you get your annotations table organized according to the way that is useful for you, if you want to push that out into a CSV file or a matrix, simply click Export Table. And we're going to call this test, and I'll push it to my desktop and click Save. Okay, it's going to be found right here. So if I double click on that, Excel will open up, and it'll look exactly the same way in Excel as it looked in Geneious. Okay, so a very quick and easy way to create, manage annotations. To delete annotations, it's also trivial. Over here in the annotations and tracks box, I can select the drop down underneath the color, and that exposes a number of options such as delete. So I can delete all motif annotations, and you'll see that they're all gone. Okay. Now, when we design Geneious, we design it for the masses, and since people use software in different ways, we've put our annotation tools in a couple of different places, but they're the same. Okay, so underneath this Annotate and Predict button, I've got a menu list of annotation tools. Okay, so find open reading frames. If you're hunting for motifs, if you are wanting to predict genes with Glimmer, if you're working with protein data, you want to predict a secondary structure, or you want to look for transmembrane domains, all of those features are found here. Okay, the same menu list is found at the very top, Annotate and Predict. Okay, so same exact list, two different places. Okay. Now, the one set of tools that I would like to really point out is this Extract Annotations tool, which is found underneath the Tools tab. Okay. When I click on that, I have the ability to extract annotations from whatever sequence data I have. So a use case might be, I've downloaded human chromosome X, and I want to extract all CDS regions. Okay, so I can define the annotation type as type CDS. I can choose what to extract. I can choose to include the 5', 3' flanking regions. And then when I click on the OK button, it will extract. Okay, so in this case, it's just a vector sequence. So let me download just for demo purposes. Okay, so this is just human chromosome Y. Okay, this is in my sample documents folder, which is found in every instance of Geneious. Okay, we have just the record for the sequence data. You'll have to actually go and download the sequence from NCBI. We have the same for bacteria, eukaryotes, plants, and viruses. Okay. Actually, this may take a little long. So let's try. Let's just try E. coli. Okay. This will show you the power of using annotations that have been affixed to the sequence to leverage. Okay. Any questions so far about what I've spoken about? Okay. Okay, so here's the E. coli K12 genome that I just downloaded from Geneious. We see we've got a pile of annotations on there. I want to extract all CDS regions, so 4,300 regions. So I just go up to Tools, Extract Annotations. I'm going to extract type CDS. Okay, and I'm going to click OK. And Geneious will go through, and it'll create a sequence list of all of those annotations. Okay, so all 4,300 annotations I extracted from E. coli in the sequence list. Okay, so just think of annotations as decorations on the sequence that impart meaning. Okay, and you can leverage that within Geneious to your advantage. Okay, so the last thing I'll speak about, shifting gears drastically, is one of our in silico cloning methods that we've added, Crispr. Okay, you will find the Crispr cloning method in Geneious underneath cloning, the cloning button at the top. The last choice is to find Crispr sites. I'm just going to speak briefly about how to navigate within Geneious to find Crispr sites. Okay, so in this case, I've got a sequence that I would like to run our Crispr tool on. Okay, so I select the sequence, go to cloning, choose find Crispr sites, and I've got this find Crispr sites menu that pops up that I want to speak about real quick. Okay, so in the first place, you can assign the target site as well as the PAM site. Okay, and if you have any questions about that, you'll click on the question mark over there to the right. Okay, I can either select or deselect on target activity scoring, okay, which will score sites through on target activity. Okay. I can score the sites through off target analysis, and this is where I can assign a database to score against an off target database. I can assign the speed and the filter strategy. And I can also designate within the speed and filter strategy how quick. Okay, and so we try to give a prompt for if you choose slow, what does that mean for you? If you choose normal, what does that mean for you? If you choose fast, okay. One of the benefits of using the Crispr tool in Geneious is that you can create your own off-target database. So the majority of the Crispr tools out there have a predefined off-target database that you have to score with. Okay, within Geneious, it could be any organism. Okay, and I'll show you quickly how to do it. Okay, with the off-target scoring database, you just simply point to a folder that contains your database of sequences, and Geneious will use that as the off-target database. Okay, so in this case, my off-target database is a human genome folder. Okay, so I've got my human genome sequence data here, all of the chromosomes that I have put into one place, and that is what I point to as my off-target database. Okay, so it's nothing more than a folder within your local database that contains sequences, and this is where we're very flexible. So if you're whatever organism you're working with, create your own off-target database. You're not limited to what somebody's already decided for you. Okay. So as I mentioned before, you have the ability to score the sites through off-target analysis. When you have defined all of the strategies for your Crispr method, Geneious will give you a status for what is happening, and then Geneious will annotate Crispr sites onto your sequence. So that chromosome 22 that we chose, we set our Crispr parameters, we pointed to the human database folder, the human genome as our off-target database. Crispr sites are now annotated onto our sequence, and they are colored according to heat map. Okay, so dark green being great, and the lighter the color, all the way to red. Red is bad. When you float over an annotation feature, a Crispr site, it will give you the metadata associated with that site. Okay, so it'll give you the off-target score, it'll give you the sequence number as well as the location interval. Okay. You can choose to color your annotations according to a number of different parameters. Okay, so you can do heat map coloring according to on-target score, according to off-target score, as well as other parameters. Okay, so if I do on-target score, then we'll see that a number of our annotation features turned red. Okay, those are bad. Okay, so I can choose to use the split panel view so that I can have my sequence view in one portion of Geneious. I can have my annotations table in the other, and I can quickly migrate or navigate through my sequence to look at all of my Crispr sites. I can even sort them. So I can sort them on off-target score or on-target activity score, whatever you'd like to sort on. Okay. Now, I mentioned that off-target database. Okay, so the off-target database in this case, I just piled a number of Vivax sequences from the Vivax genome into my folder, and that's what I point at when I run this CRISPR method on my sequence data. Okay, so it's the user has freedom to decide what they would like to use. Okay. Okay, so with that in mind, I think that brings us just up to an hour. That's quite a bit of time to talk at a very high level. I'd just like to take the opportunity to thank everyone for their time today. I'd like to save some time for questions. I'd also like to give you my email address. So if you'd like to contact me offline, I'm happy to do it. I'm up in Boston often. So while I'm up in Boston, if you'd like for me to pop in and sit down and work with you, I'm happy to do that. But just know that you can reach out to me. I'm available and I'm happy to help. So thank you everyone for your time. And please let me know if I can answer any questions.
Great. Thank you very much, Christian. So if anyone out there has questions, feel free to chat them to me. You can chat to me here in the chat window. We have a question from our local group here. So go ahead.
Yes. Hello, sorry to ask you. I like the annotation automatic tool for finding restriction sites. So do you have some automatic tool? Maybe it's a plugin or something that you can find when you have a DNA sequence, you can find in the translation some motifs like the TEV cleavage sites, protease cleavage sites, or like flags. Is there a plugin or tool that you can automatically find this, annotate these sites?
Yes, yes. So there are two tricks. One of them, and it's kind of two ways to skin this cat. You can hit the Control F button, and you can find a motif within your sequence viewer. Okay. Now, in this case, I'll just type in ATG because I know there's a ton of them. So I'll find them all. So Geneious found 76,000, of course, and I'll click OK. And Geneious will select all of those ATG sites in my sequence viewer. Okay. So that's one way you can do it. It'll select the sites and then you can annotate them all at once. The second way to do it is underneath Annotate and Predict, there's a Search for Motifs field. Okay, now we use sequence or prosite motifs. And in here, we actually use the emboss fuzznuke method. Okay, if you're not familiar with that, you can click on the fuzznuke link and it'll open up the browser and give you documentation for that. But this is also another sweet way that you can look for motifs that you know about. This will also accept regular expressions. So if your motifs need to be flexible, such as, you know, first 6 positions are absolutely this, but the 7th position could be this or this, you can include that here. And when you select the OK button, it will highlight, if it finds the motif, those motifs in your sequence. Okay, so 2 ways that you can find motifs. The quick and dirty way is that Control F. Okay, you can just plug in your sequence. Now the Control F does not take regular expressions, so that's the one limitation of this. But when I'm looking for you know, motifs of whatever type, this is usually my first place that I go just to do a quick search. If I know they're there and this doesn't find them, that's when I drill down into search for motifs.
Okay. So, sorry if I mean, I'm not very much aware of the second way that you saw, but basically to refine most of the question, it would be very useful if you could make like a small database of 5 or 6 motifs that you are regularly searching for, and you can always be searching for this set of motifs, for example.
Okay, so you can do that as well. So let me point out a tool. So here I've got my E. coli genome. This orange tab over here to the right, this is where you can annotate from. So if you have a folder, and I'll just create a folder of motifs. I'll do a new folder, and I'll call this motifs. Okay, so in this folder, I can put however many motifs I want. If I would like to see if my sequence of interest has those motifs, I can simply point to that folder. Okay, so I'm going to, let's see, this is underneath genomes, bacteria. So I'll point to the motifs folder. I'll select OK, and any motif that is found, and this actually is a really nice way to do it because it uses pairwise identity. So it'll offer you a little bit of flexibility with those motifs. So you could have, 10,000 motifs in here, point to it from your sequence of interest. Over here to the right, the annotate from, and I'm just pointing to that motifs folder that I have shown here. That is a way you can also search for motifs. So that might be helpful too.
Great, thank you. One other question, that I said, the workflows are really powerful, and their portability and shareability is fantastic. That would seem to lend itself to some sort of community resource where you could sort of have a hub of sorts where people could deposit and extract and modify workflows. Is there anything like that in the mix or on the drawing board?
Yeah, so that is on my list of things to do for version 10. This is something I thought about one version ago, but it's not trivial to do. To set up a workflow community as a resource that you could go to our Geneious support site, look up NGS workflows, download a workflow, and then tweak it. Okay, so that's my grand scheme to take over the world. For the time being, what I've encouraged, especially our commercial users, is that you'll have cloning groups or protein production core groups or NGS groups, is to start depositing their workflows in one space. And then as an internal resource, that's a way you can do that quite quickly. But for a global Geneious user base, my intention is to help develop or help build out a workflow resource. So SB Grid users, hard users can go to the site, download, and run with it.
Yeah, that could be, I think that, you could back it with something like GitLab or some Git-based things, so you could do it in GitHub, and
Yes.
you could push and pull from Geneious, which would be spectacular. Be
Absolutely.
really
Yeah,
all
absolutely.
right. Well, with that, I think we'll leave it at that. Christian, thank you very much for the great webinar, and I'll follow up with you offline. And thank you, everyone, for joining.
Okay, thanks so much.